So with the background of the citric acid cycle behind us now and having a general sense of what we're going to be doing in these reactions, we can proceed and take a little bit more of a detailed view into this set of reactions specifically. So we're still in the first half of chapter 19 here. We're still setting the stage for the citric acid cycle, but going a little bit more in detail. We brought ourselves up to speed with glycolysis first thing, and then we talked about the role of the citric acid cycle generally in metabolism, kind of setting the stage for what we're about to do in the citric acid cycle. What we'd like to use this chunk for is talking about the citric acid cycle and its reactions from the bird's eye perspective. Before we get into the nitty-gritty of the chemistry, I kind of want to give you the punchline before I tell you the joke and let you see where we're going. Much like we did for glycolysis, once we have a roadmap, we can proceed ahead in earnest. Once we do have that roadmap in hand, we're going to back all the way up to pyruvate where we started, and we're going to talk about the biochemistry of converting pyruvate into acetyl-CoA and go through that process in a pretty detailed view. And then in the last lecture chunk, once we've made acetyl-CoA, we can talk about the beginning, about the first half of the citric acid cycle itself. I think we'll cover about 4 reactions at the end of this lecture chunk. So here it is in all its glory, the citric acid cycle. We start with pyruvate, we convert it into acetyl-CoA, and no surprise that this is a cycle. We're going to go around and come all the way back to regenerate our precursor so that we can repeat this process again and again. Once we make acetyl-CoA, and remember, we'll talk about the biochemistry of making acetyl-CoA by the end of this chunk. Once we make acetyl-CoA, we link acetyl onto the first intermediate of the citric acid cycle called oxaloacetate. CoA is recycled. It goes back to be linked to another acetyl molecule way up in the precursor, the previous step here. But the acetyl that we have made will be linked to oxaloacetate. When we link acetyl, which is 2 carbons in size, to oxaloacetate, which is 4 carbons big, we create a 6-carbon molecule called citrate. Citrate is isomerized to isocitrate, and then we have 2 oxidative decarboxylation reactions. These are reactions where we steal electrons from isocitrate. By stealing electrons, we're oxidizing it, and a net result of the oxidation events are decarboxylations where carbon dioxide is released. So we'll do 2 of those in a row. 2 successive oxidative decarboxylations in a row, each one of them stealing electrons and, more importantly, perhaps causing carbon dioxide to be released. These are the 2 carbon dioxides I told you in the last chunk we would be releasing. At the end of those 2 reactions, we're left with a molecule called alpha-ketoglutarate, perhaps the most important citric acid cycle intermediate. Alpha-ketoglutarate is used to make many different amino acids, getting back to that role of the citric acid cycle in anabolism. But as we continue on in the cycle, alpha-ketoglutarate is made from the first of those 2 oxidative decarboxylation events. The second oxidative decarboxylation results in a molecule called succinyl-CoA. Succinyl-CoA pretty instantly becomes succinate, with the CoA molecule being released, and then the rest of the citric acid cycle is truly for cycling, is to bring us back to oxaloacetate, where we can repeat this process again. Along the way, we'll see that we make 1 molecule of GTP, guanosine triphosphate, in the citric acid cycle. Big deal. We actually use that single GTP to phosphorylate ADP to ATP, so it's a very quick conversion, where the GTP molecule we just made is used to fuel the phosphorylation of an ATP molecule. Well, big deal again. I mean, 1 ATP molecule for all this trouble. It's not really what we're after. And it's not the point of the citric acid cycle. The citric acid cycle doesn't exist to give immediate energy payoff. It exists to harvest electrons for later payoff, for ATP production that's going to come later on. But one thing I hope here is obvious. We can't do anything with pyruvate with regards to the citric acid cycle. The very first thing we need to do is convert that pyruvate into acetyl-CoA. So let's go ahead and get to it. Let's dive into the true biochemistry here. Let's start with pyruvate, which we have made through glycolysis, and convert it into acetyl-CoA that we can feed into the citric acid cycle. So the first thing we have to do is get the pyruvate out of the cytoplasm, which is where glycolysis lives, and get pyruvate into the mitochondria, which is where the citric acid cycle will be. And that's a pretty trivial matter because pyruvate has its own mitochondrial transporter, its own mitochondrial membrane protein that brings it into the mitochondria. Once pyruvate has been transported into the mitochondria, it is acted upon by a huge enzyme complex named the pyruvate dehydrogenase complex, the PDC. And know this complex inside and out. This is going to be one of our best friends in this unit. We will know this complex intimately. We talked about dehydrogenase enzymes before. They kind of sound like they steal hydrogens, and in fact that's indirectly true. Really what dehydrogenase enzymes do is steal electrons, and the protons follow. So this is going to be a redox enzyme. This enzyme is going to catalyze a redox reaction for us. A little bit more specifically, the PDC will first decarboxylate pyruvate and then steal electrons from it, converting that pyruvate into acetyl, and the acetyl will then be linked to CoA in an activation step in order to make that acetyl more reactive so that it will willingly enter the citric acid cycle. Like all things linked to CoA, acetyl will link to CoA through CoA's thiol group, creating a high energy bond, and that energy will be what fuels the condensation of acetyl with oxaloacetate later on. To do this, to convert pyruvate into acetyl-CoA, we have to have a few different steps, and actually we need different enzymes for each of those steps, and luckily for the cell, luckily for us, each of those individual enzymes are subunits of the PDC. So the PDC is big, it is multi-active, it has many different activities, many different enzymes that are part of it, and it is incredibly impressive. Also, the reaction of converting pyruvate into acetyl-CoA is an exergonic reaction, and it releases about the equivalent of an ATP molecule's worth of energy, about 33.5 kilojoules per mole. So here it is, a little bit stylized, but this is an accurate representation of what the PDC probably looks like in the cell. The colors are false, of course, and you can see that it's beautiful. It is just a beautiful, beautiful enzymatic complex. There are 5 different types of enzymes that make up this complex. It takes 5 different enzymes to both perform the catalysis of the PDC and regulate it. 3 of the enzymes that we find in the PDC are actually needed for the conversion of pyruvate into acetyl-CoA. The remaining 2 are regulatory enzymes. One is a kinase and one is a phosphatase, and together they're responsible for turning the PDC on or off depending on our energetic needs in the cell. So that leads us to realizing that the conversion of pyruvate into acetyl-CoA, the action of the PDC itself, is a control point in glucose metabolism. We can turn metabolism up or down. We can increase or decrease our ATP production by regulating the activity of the PDC. Converting pyruvate into acetyl is going to involve another coenzyme that we haven't met yet called lipoic acid. Lipoic acid is an interesting coenzyme because it is not only important for activating non-reactive molecules, but lipoic acid is an oxidizer itself. It can steal electrons. It can cause itself to be reduced and steal the electrons from things that you link to it. And that's going to be important for us in just a moment as well. So this is probably the most informative figure from this lecture chunk. This essentially summarizes the entire activity of the PDC in one nice flow diagram. If you could recreate this or extract information from this, you'll be set for any information that I might ask you about this material on the upcoming exam. So let's talk our way through this, always using this figure as our roadmap for the PDC's activity. Step 1 of the conversion of pyruvate into acetyl-CoA by the PDC is catalyzed by an enzyme subunit called pyruvate dehydrogenase. Pyruvate dehydrogenase decarboxylates pyruvate. Right there in step 1, we release a carbon dioxide. That means we've gone from 3 carbons to 2, and what remains, what's shaded in blue here, referred to as a hydroxyethyl group, is linked onto a coenzyme called TPP. TPP stands for thiamine pyrophosphate. You'll never need to know that in my class ever. So what we've made at the end of step 1 is hydroxyethyl TPP. Essentially, again, to summarize, pyruvate is decarboxylated, the carbon dioxide is removed, the hydroxyethyl group that remains is linked to TPP as an activation step. Step 2, moves hydroxyethyl onto lipoic acid. The enzyme responsible for this is called dihydrolipoyl transacetylase. I will not ask you for the name of that enzyme ever again. Again, this enzyme transfers hydroxyethyl to lipoic acid, to the coenzyme lipoic acid. That you do need to know. So step 2 of this conversion is moving hydroxyethyl from TPP to lipoic acid. This step requires that coenzyme lipoic acid, no surprise, because that's what we're linking hydroxyethyl to. And lipoic acid is going to do what it does best as a coenzyme. It is going to steal electrons. So lipoic acid becomes reduced, stealing electrons and protons from hydroxyethyl, and hydroxyethyl has just been oxidized. Now here's the interesting thing. Oxidized hydroxyethyl ain't hydroxyethyl anymore. Oxidized hydroxyethyl is acetyl. So by the end of step 2, we are left with acetyl linked to reduced lipoic acid. Again, to recap, once lipoic acid steals electrons and protons from hydroxyethyl, hydroxyethyl converts to acetyl. So we have acetyl linked to reduced lipoic acid. Step 3 involves the CoA enzyme coming in and attacking the bond between acetyl and reduced lipoic acid. Essentially, CoA steals acetyl, resulting in acetyl-CoA. So we've just made acetyl-CoA. The enzyme subunit of the PDC that catalyzes that reaction is called dihydrolipoyl transacetylase. You don't need to know that either. Same enzyme as we mentioned before. All that's left, the remaining reactions of this conversion, are not to make acetyl-CoA. We just made that. We're done. It is to recycle the enzyme back to what it was before so it can perform this catalysis again. Step 4, lipoic acid is oxidized back to its original form, and it is FAD that then takes the electrons and protons that had previously reduced lipoic acid. FAD becomes FADH2. The problem with FAD is that it is not a mobile electron carrier. FAD really can't go anywhere. So FAD then offloads the electrons and protons to NAD, and NAD brings those electrons and protons to the electron transport chain. Once this is done, step 5, the reduction of NAD to NADH plus H, everyone is regenerated and the enzyme is ready to repeat the process again. So the overall conversion of pyruvate into acetyl-CoA is summarized here in its most simple and straightforward way. We start with pyruvate and we end with acetyl-CoA. But along the way, CoA went in, carbon dioxide came out, NAD plus went in, NADH plus H came out, and we needed the PDC itself, we needed TPP, we needed FAD, and we needed lipoic acid. We don't discuss the role of magnesium, just to keep it a little bit simpler. So overall, when pyruvate is converted into acetyl-CoA, we've harvested 2 electrons and transferred those eventually to NADH plus H, where they can go to the electron transport chain. These are actually our first electrons that we've harvested from the metabolism of pyruvate. So the citric acid cycle is already hard at work harvesting electrons, and we haven't really even entered the citric acid cycle yet. These electrons will eventually be used to make ATP as part of the electron transport chain. Now the pyruvate dehydrogenase complex is truly an awesome complex to behold. It is a package deal, literally in a single package. Everything you need for converting pyruvate into acetyl-CoA and regulating that conversion from an energetic perspective is all in the one complex of the PDC. And not only that, it is very organized and streamlined. You start with 24 individual subunits of dihydrolipoyl transacetylase. Remember that enzyme we use in 2 different steps of the conversion. They are arranged as 8 trimers in a cube-like orientation, and we see them here on the left, kind of in this dull purple. If we think of that as a cube, along the edges of the cube are another 24 subunits of pyruvate dehydrogenase, the enzyme that we need in step 1 of this conversion, the enzyme that decarboxylates pyruvate into hydroxyethyl. Those 24 subunits are arranged as 12 dimers, again, along the edges of the cube. On the faces of the cube are 12 dihydrolipoyl dehydrogenase enzymes. These are those enzymes that are responsible for regenerating lipoic acid. These 6 dimers are on the faces of the cube. This arrangement allows something that is sometimes referred to as substrate streamlining or substrate feeding, where substrate as it leaves one enzyme as product is perfectly positioned to enter the active site of the next enzyme as substrate. You can think of this as the molecular equivalent of Henry Ford's factory line, where as soon as one enzyme works on a substrate converting it into product, that product is right there to be the substrate for the next step. Huge improvement on the speed and efficiency of the enzymatic catalysis, and it allows for potent regulation because all of the enzymes involved in this process are there in one spot. So to summarize what we've just done, we've converted pyruvate into acetyl-CoA. We took the pyruvate that we've made at the end of glycolysis and we fed it into the PDC. The PDC went ahead and decarboxylated that pyruvate, making a molecule called hydroxyethyl. Hydroxyethyl was linked to TPP, giving us hydroxyethyl TPP. We then moved that hydroxyethyl from TPP onto lipoic acid, and as we did that, lipoic acid stole electrons and protons from hydroxyethyl, and that oxidation caused hydroxyethyl to become acetyl. So now acetyl was linked to reduced lipoic acid. CoA then swoops in and attacks the bond between acetyl and lipoic acid and steals acetyl, giving us acetyl-CoA. The last 2 steps are for the regeneration of the PDC itself. First, FAD has to take the electrons and protons from lipoic acid, restoring lipoic acid to its oxidized form, and then NAD takes those same electrons and protons from FAD and restores FAD to its oxidized form. NAD can move those electrons to the ETC, the electron transport chain, because NAD is a mobile electron carrier. So quite a lot goes on in the conversion to acetyl-CoA, but we are now there. We've made acetyl-CoA, 2 molecules of acetyl-CoA for every molecule of glucose consumed, and we are poised and ready to feed that acetyl into the citric acid cycle, and that's exactly where we will pick up in the next lecture chunk.